Thank you, Roger, for the invitation. I'm very glad to present today. Today, the presentation is both like to bring interest into the RNA binding proteins. It was mentioned a bit yesterday that EV takes mostly all the attentions in the area of exRNA, extracellular RNA. But RNA binding proteins are, I would say, gaining interest in understanding the biology of exRNA stabilization and sorting in cells. And that's basically our main focus as part of the ERCC2 and also in the lab. So I would just go very quickly on the major challenge that we have and why we are interested into exRNA and also RNA binding proteins for biomarker studies and also for understanding the fundamental mechanism releasing extracellular RNA in biofluids. One of the major problems in diagnostics, and people mentioned this yesterday, is the dilution effects of the disease-relevant biomarkers in the overall exRNA circulating from different organs. And the second challenge is the diversity in the exRNA carriers. We learned from the ERCC1 and many other publications that circulating RNA are embedded into really a plethora of carriers ranging from single proteins like RNA binding proteins to lipoproteins ranging from a couple of nanometers to several microns. And also all range of extracellular vesicles, exomer, maybe also RNA granules, as well as organelles such as mitochondria, and of course, viruses that could circulate RNA during infections. Today, we are going mostly to speak about RNPs or RBPs, depending on how we want to call them. And why I was getting interested in this is mostly from my initial conversation with Kendall Jensen a long time ago, when she showed me the different profiles of exRNA in different biofluids. And as you could mention, there is huge variation in RNA biotype abundance in plasma, saliva, or urine, where urine, for example, most of the RNA is composed of tRNA, while in plasma you can see microRNA and yRNA, for example. So why there is such a big difference in RNA biotype? And one hypothesis would be that there is difference in RBP composition. And second hypothesis would be that different RBP stabilize or sort specific RNA fragments or biotypes such as microRNA and RNA in the extracellular space. It's not new. I mean, there is many RBPs that have been described as stabilizing or sorting RNA in the extracellular space. And I would like to advertise this nice review that was recently published in GEV last year, where I would say a couple of, maybe a dozen of RBP has been carefully characterized. But the problem is that it was in different publications, different teams, and there was no coherent analysis and review about the impact of each of the RBPs. But this is still a proof of principle that RBP can impact exRNA profiles. How we could do that? There is different hypotheses, notably the fingerprint hypothesis. We talked about it yesterday, where an RBP will protect RNA fragment from degradation in the extracellular space, leaving a kind of clip, and that's we are going to speak about clip signatures in the RNA sequencing. Otherwise, there is all kind of sorting or passive or active sorting that could occur such as RBP that goes to specific subcellular space compartment, RNA editing by RBPs that could stabilize or sort the RNA, exRNA sorting by binding, for example, to membrane-bound adapter protein. There is several examples about this. Organelle enrichment, for example, if there is RNA binding proteins stabilizing specific organelles, they will of course stabilize organelle-specific exRNA. And also possibly because we know that RNA binding protein assemble into multi-layer, nearly micron size RNA granules, they could also stabilize RNA in global scale this way. So that's why I represent here EV extracellular organelles or granules that could be the carriers and composed of RNA binding proteins. So the current challenge to identify and characterize exRNA binding proteins are the heterogeneity of the experimental systems. All publication has been described in different model cell line, different techniques to analyze the impact of RBP and sequencing methods. So there is no currently study that studied the impact of each RBPs independently. Also, there is, as mentioned just before, there is a lot of RBPs, nearly 1,000 in the human genome, and beyond candidate approach and serendipitous discovery, it's pretty hard to analyze the impact of each RBP using an unbiased approach. And also there is technical difficulties to characterize RBPs in biofluids. They are in low concentration and mixed with very complex mixtures, and they could be associated with EV or non-EV. So we need to develop new approaches to characterize these RBPs exRNA interactions. So it's good that I had the chance to talk after the Eric's talk. We have been, of course, fascinated by the study they have performed on ECLIP and cataloging the impact of xRBP. And we are combining this approach with mass spectrometry predictions to identify high probability, what I would say high probability exRNA binding proteins. And we are validating these candidates using CRISPR engineering and immunoprecipitation in parallel. So this bring me back to this idea or hypothesis of exRNA RBP fingerprints, where we can actually combine the ECLIP data and exRNA profile data, taking the hypothesis that RNA binding proteins are protecting RNA from the degradation and just leave a mark where they are actually associated with the RNA fragments. And we collaborated with Jean a couple of years ago, where he told me about this effort, and we start developing tools to identify RBP in the extracellular space based on exRNA profiles and ECLIP profiles. So the idea of what we call the exRBP bioinformatic pipeline is actually pretty simple. It's basically an intersect of different peak calling from the exRNA profiles and ECLIP profiles from different sources, and most of it is the ENCODE profiles. Then we run permutation analysis to just calculate p-value and just test whether this co-occurrence of RBP, ECLIP profiles and exRNA profile are just occurring by chance, or just because there is a lot of peaks on the ECLIP data, or a lot of peaks on the exRNA profiles. And then we restrict this list using p-value and rankfold change to identify the one we select for downstream validation. So this is just a heat map showing some analysis in different biofluids such as plasma, saliva, and urine. This is not extensive, and this one actually don't represent the permutation analysis, but just to give you an idea of what kind of information you can extract on nearly 150 RBPs in 2 cell line, which has been used for the ENCODE data. What we can also extract is XLBP associated with a specific RNA biotype, such as microRNA, SNO-RNA, snRNA, volt-RNA, and yRNA. We can deduct interesting associations, such as, of course, the known association with GCR8, DROSHA, and other RNA silencing factors with microRNA, and TROF2, for example, with yRNA. And this is not extensive. Most interestingly, the RBP we identify, for example, with mRNA fragments that people mentioned over the last session. For the experimental validation, what we do is we combine CRISPR engineering on a model system, which is 293T, and immunoprecipitation to validate the hits that we identify through the pipeline. Just to give you an idea of what we can achieve, I just present you a case study on RNA silencing protein pathway. RNA, microRNA pathway are processed and stabilized by a series of maturation and stabilization, including DROSHA, GCR8 in nucleus, sorting into the cytoplasm by exporting run factors, and then a second cleavage by di-30-RBP, stabilization in argonaute proteins, and stabilization also in the extracellular space by different argonaute proteins. So we generated a collection of isogenic RNA silencing knockouts, all in the same background, just to be able to compare side by side the impact of each inactivation. And generated basically one first atlas of I would say, RBP impact on extracellular RNA. So where we can analyze the impact of each inactivation in the cellular RNA profiles and the extracellular RNA profiles. The idea is to generate this heat map for each of the RBP that we identify for the pipeline, and then basically growing the list of RNA binding protein that we can identify. So here is, of course, the impact of RNA silencing proteins on microRNA is obvious. We can, of course, see the impact it has on extracellular microRNA accumulation. For example, if we inactivate argonaute or the key maturation enzyme. What is also very interesting is looking at the impact of inactivating, for example, extracellular microRNA or cellular microRNA on the stabilization and export of other biotypes such as tRNA, snRNA, and SNO-RNA. I mean, I don't want to make a whole study and catalog. I'm actually already running out of time. I just want to advertise this work and mention that we are willing to collaborate on this if people are interested. I think I will just stop there. So just in summary, we believe that RNA binding proteins are major players in exRNA stabilization and sorting. We developed this pipeline allowing to exploit the ECLIP data and exRNA profile data to identify high probability exRNA binding proteins. And we are currently establishing a first functional exRNA binding protein atlas to define the respective impact of each RBPs on exRNA profiles. So we are currently, we have finished phase 1, which was around a 14 RBP combination. We are nearly done with phase 2, which is now 35 RBPs, and we'll start phase 3 from September. So I would like to thank the team, and notably Alessandra Sturtler and Veronica Barragan-Boero, which were key members who generated knockouts and characterized exRNA profiles. And also would like to highlight my collaborators in teaching the Bunker and Jensen group, and Rafael group and Demelo group in DH, which we collaborate together for the PRISM project, but from the ERCC too. Thank you very much.